have a seat at their stool, and I want to welcome you to Nighthawk Biotechnology. So this is where, at Ironwood Ridge High School, we have our Bioscience program. So my name is Mr. Pincus, and I teach the Bioscience program. I've been teaching at Ironwood for, this is my 4th year, and prior to that, I worked in industry for 20 years, where I did drug discovery, research, and development. So today, what we're going to do in our lab is we're going to do an experiment that we usually do with our Bioscience 1 students. You are going to isolate your own DNA. You're going to isolate your own DNA from your cheek cells. So I have 2 students with me. I'm going to allow them to introduce themselves. They'll tell you what year they are in the program, and they're going to be the ones that are going to lead you through this experiment.
All right. My name is Nick. I'm in year 2 of the Bioscience program.
And I'm Leah, and I'm in year 3 of the Bioscience program.
Okay, so let's go ahead and get our experiment started.
Okay. So for our very first step, I'm going to have you go ahead and locate on your lab benches the 15 ml tube. That's the clear one with the blue cap. And it will be containing water, so it won't have any sort of labels on it.
Okay.
So you'll find one without a label on it, and go ahead and put your initials on it with the Sharpie provided on your desk. And once everybody has that done, you will go ahead and put the tube back in the green rack where you found it, and then you're going to chew the inside of your cheeks for 30 seconds, and drawing blood won't be helpful, so just kind of gently break apart the skin that's on the inside of your cheeks and on your lower lip, if that's helpful.
You're going to want to take that tube, unscrew it, the one with the water in it. You're going to want to take that tube, unscrew it, and swish the water around in your mouth for around 30 seconds. So this is what we call a transfer pipette. Find the tube on your bench labeled lysis. It should be the other big tube on there. So what you're going to want to do with that is draw 2 milliliters out of it and put it into the tube that you just spit the water back into.
So we're going to take the tube that you just put the lysis buffer into, the one with your initials on it. And you're going to do what's called inverting 5 times. So you're going to make sure it's screwed nice and tight, and you're going to flip it up and down just like this 5 times. So then next what we're going to do is you're going to see a colored microcentrifuge tube. It'll be different based on each table. So like for example, this table has a green tube. So if you find a little microcentrifuge tube that's colored, what we're going to do using our transfer pipette is we are going to add 5 drops of that solution into the tube that you just mixed with.
All right, and so next what we're going to do is we're going to take your tubes and put them in this water bath for about 10 minutes.
So what kinds of questions do you guys have about what it's like to take a bioscience class? No one's asked when you can even take it. What do you got?
Does Bioscience have credits for college too?
It does have credits for college. So when you take Bioscience 1 and Bioscience 2, you can actually earn credit to go to that goes through the University of Arizona. One of the biggest and best projects we do in this class, in Bioscience 1, is we actually make bacteria glow in the dark. So the students actually do all of the work to change the genetics of the bacteria to actually cause them to glow in the dark. It's a lot of fun. When you move into the more advanced classes like Bioscience 2, you actually get to use that piece of equipment which is behind me. This is called a tissue culture hood. And inside of this, we culture human cells. So we'll actually have cells that are in that freezer right over there that we'll bring back to life, and then we'll do experiments with those cells. If you come in as a freshman and take bioscience, you can actually do Bioscience 4 or Bioscience intern. And when you're an intern, that means you get the opportunity to do research with me, probably through the University of Arizona, because I have some colleagues at the university that we're going to do some research with, and you get to help teach the Bioscience 1 class.
What's the difference between CTE and emotional? What's the difference between the classes, different classes?
Okay, that's a really good question. So CTE classes are 60% hands-on at a minimum. So that means you're doing work with your hands in a laboratory-based environment, like with this class, it's going to be in this room. 40% of the time, my classes are in that other room that you were in when you first walked into the space today. In that other room, that's where we do lectures, and we actually prepare to do the laboratories. That way we have our knowledge, and then we come in here and we put it to work. There's some right at the top there. We're going to go like this. See that stuff that's rolling around? That's your DNA. Okay. See how it's knotty? See how it's sticking there?
Yeah.
Okay. So now we can put it in there. Perfect. Go ahead and cap it. And then, Nick, after you're done with the group, just take this whole tube right into biohazard. And make sure the tip goes into biohazard also. Okay, pretty cool.
Yeah, thank you.
All right, let's do yours now. See how it's all right here? Try to get that up. How it's sticking to the side of the tube. It's because it's very viscous and it's not. Close that up. All right. So everyone has their own DNA now. So when you go back to class tomorrow, you can actually show on Zoom your DNA to your teacher. And you can say, this is what I did over at Ironwood yesterday. So go ahead and put the string through kind of the little loop that is on the jewel, and you can put it around your neck so that you have your DNA with you until you go home. And then you can give it to your mom or dad. Yep. Okay. Good work, guys. All right. So you guys can head downstairs. We're going to be in Mr. Hauser.